Well, so welcome everybody. Thanks for showing up here bright and early on a cold Wisconsin morning. I have the distinct pleasure of getting to introduce Expery Omollo for his thesis defense seminar. So Expery is a PhD student who came to my lab about 3.5 years ago and has been incredibly successful in getting through graduate school. It's customary to introduce people and give a little bit of background and sort of say something about, what is unique about them. So I will try to do that and what Expery's unique contribution to our lab has been. So Expery grew up in Kenya, went to high school in or around Nairobi and graduated there around 2016, 2015. Came to the United States to go to college at Michigan State University, our sister school in East Lansing. Expery told me that he chose Michigan State because he thought it had a great sports reputation, and he didn't know a lot else about it, but didn't know he was gonna move into the bitter cold of Michigan. So he had a unique transition in coming, finding out about the U.S. Came to Wisconsin immediately after the pandemic, fall of 2020, and Expery's eagerly looking forward to going to his graduation ceremony here because he never got to go to a graduation ceremony in the spring of 2020 when the pandemic was in full blown. So Expery has been a really productive graduate student, and I think what sort of stands out to me most is sort of a unique characteristic of Expery. Every student, I think, as you have a number of students go through lab as I have, you sort of learn that every student is different in some way, right? And takes a different trajectory, has different characteristics, even though they're all sort of there to learn how to do science. And so one of Expery's really defining characteristics is he likes to ask sort of impertinent questions. And I can remember early in the time he was here, being down at Wisconsin Union with the whole lab, sitting around, having a drink, and Expery decided this is a great time to sort of quiz me on where did all the lab's money come from and what was I doing with it all? And the students, I think, who were there with us were a bit aghast that, this was an impertinent thing to be bringing up, but actually very characteristic of Expery to sort of ask questions like that. And I think, not just about money, but I think one of the things that was quite enjoyable for me, I meet with students periodically through the time they're like every other week, we have an hour set aside. And actually often would show up and just have a question like, can you explain to me this thing that I don't understand? Or we're trying to publish a paper, I don't understand how the math works for this particular thing. Can you explain it to me? And be very insistent that he got an explanation that he understood. And I think that's, a really good characteristic of any scientist. Ask impertinent questions and stick with it until you know the answer. So I have really appreciated that about Expery. I think the other thing that's certainly merits comment, Expery went to graduate school in 3.5 years, and people look at that and think, well, how could that possibly be? Most graduate students take 4.5 to 7 years. It's a wide range. It's unusual for a student to get through that quickly. And I think the answer is really two things. One, Expery just worked incredibly hard for like 2 years when he was in graduate school. He worked basically nonstop, and that is atypical. And it was impressive. One of his committee members, Liz Campbell, is here. We often were doing collaborative Zoom calls during that time, and it was really striking how we'd have one meeting and talk about something, and then we'd have another meeting the next week, and Expery would show up with all these experiments he had done, which nobody actually asked him to do. He would have gone off and tried to figure something out, and it was quite impressive that somebody was working that diligently. So that's half of the answer, I think, that he just worked really, really hard for that short period of time. 2 years, I guess, relative short period of time. The other answer is, Expery collaborated really extensively. He collaborated with a group at Rockefeller. He collaborated with a group in Shanghai. science in the 21st century, to be productive, is necessarily highly collaborative, and you can be much more productive by being collaborative. So I think it was really those two things put together. Expery was able to get a lot done in a short amount of time, and I think he's going to now tell you about what he got done, and you all can look forward to a really interesting and entertaining seminar. Expery is a great speaker, so I think we can look forward to that.
Thank you. Well, thanks for the introduction, Bob. And I'll take the role and guide you through what I've done over the years, and hopefully you get something out of it. So as you've heard, my name is Expery Omollo. I'm a PhD student in Bob Landick's lab. And I'll go ahead and start with my talk by introducing a common disease called tuberculosis that most of you are familiar with. So tuberculosis, what we know is it causes over 1.5 million deaths each year, and it's been around for approximately 3 million years. So this is as long as we've known the human history. And the challenging thing about this tuberculosis is we haven't figured out how to completely get rid of it. Now, we've had treatments that currently target this tuberculosis, and they usually target the pathogen that causes the TB directly, okay? And with that said, I'll introduce what the pathogen is. It's called Mycobacterium tuberculosis. And the way the pathogen works is when you have an infected lung like this, you have this tiny bacteria that invade your lungs and makes it harder for you to breathe, all the symptoms that are known for tuberculosis. And although we've been able to treat it, recently there's been a lot of increase in drug-resistant MTB strains, which has caused an approximately 44% drop in the global cure rate. So this is a problem because if we let this keep happening, it's gonna get so bad that we wouldn't be able to treat tuberculosis at all in the future. So with that said, we definitely need to develop alternative drug candidates in order to fight TB. And where I come in with my project and with our group is we were asking, can we target the transcription machinery in Mycobacterium tuberculosis in order to find alternative drug candidates? That was the main question that led to this research. Now, I'll introduce what transcription looks like for most of you who may not be familiar. This is an overview of transcription. And the process itself usually starts when the holoenzyme or RNA polymerase enzyme recognizes the promoter sequence here, and then it initiates transcription, which transitions into transcription elongation over here. So during transcription elongation, sometimes the RNA polymerase can pause for some regulation to happen, but if there's no regulation, it goes at full speed until it reaches the end or the point where it needs to terminate transcription. At this point, the RNA that was being synthesized is released, and the polymerase that was synthesizing it is also released, and this polymerase is recycled, and this process goes on and on until all genes in the cell are transcribed. Now, for my talk today, I'll be talking about two parts. And one will be focusing on the interplay between transcription elongation and how this pausing regulates transcription. And the second part will primarily focus on what happens during transcription termination. And so I'll dive into part one. Here, I'll be studying transcription elongation in Mycobacterium tuberculosis. And before I talk about it, I'd like to introduce the enzyme RNA polymerase and exactly what it does, okay? So what I'm showing you here is one enzyme, RNA polymerase, that is colored differently. So the different colors just tells you that this is a multi-subunit polymerase. So it's a multi-subunit protein. The parts we're interested in is the RNA in red that is being synthesized, and the DNA is what determines the sequence of this RNA, okay? So those are the only three things you need to know from this complex. So how does it work? The way it works is you usually have an incoming nucleotide over here, that comes into the active site and gets incorporated into this growing RNA chain. And once it gets incorporated, we call this state the pre-translocated state. And then you have translocation happening, and we now call it the post-translocated state, okay? That is all you have to know about transcription elongation in bacteria as well as eukaryotes. Now, with that said, transcription itself inside the cell is extremely fast. And we know, at least in some bacteria, this can take place at a speed of 30 to 100 nucleotides per second. Now, my question to you, the audience, is if you wanted to slow down this polymerase or to stop it, like how will you slow down such a fast-moving RNA polymerase? And I'll tell you how. So we know there's a mechanism that's been described for how the RNA polymerase can slow down, and it's been referred as elemental pausing. So what is elemental pausing? These are short pauses that the RNA polymerase usually makes during transcription. How does it work? It usually works by scanning signals that are encoded on the DNA and RNA. So these are highlighted in yellow over here. But once it sees those signals, it tells the polymerase, hey, you need to stop at this particular position. So then what makes this polymerase stop is actually you have to pay attention to what's happening in the active site here. You'll notice the RNA in red is shifted forward, but the DNA is left in the old position such that it's making this weird slanting hybrid between the DNA and the RNA. That is because the RNA has translocated forward, but the DNA failed to translocate, okay? And just to make sure that you get the point, I'm gonna show you another animation for how that happens. So here you have an incoming nucleotide that comes into the active site, gets incorporated, and then you have the pause signal. You have the RNA translocating, but the DNA fails to translocate. The transcription cannot go on in this scenario, and we call that elemental pausing. Now, why will RNA polymerase pause in the first place? You may be wondering. So there are some published reasons, but there are way more reasons that I won't talk about today. But one of them is usually to allow formation of secondary structures in the exit channel over here, like this guy over here. And sometimes is to facilitate coupling between transcription and translation. Sometimes it acts as a prerequisite for transcription termination. And sometimes it can also prevent collisions with other proteins that are bound on the DNA, okay? So, but for my talk today, I'd like to narrow down to this particular event here. Like, how does the initial elemental pause allow RNA structure to form here? And what is the role of this RNA structure? So with that, I'll introduce this RNA structure to the right. On the left, this is what I just introduced. And this RNA polymerase pauses here, and I want you to pay attention to the half-life of this pause. This pause will take about 15 seconds, and this 15 seconds is sufficient to allow the RNA here to start folding onto itself and form this weird structure here. One thing you'll notice is this structure resembles a hairpin structure. And so that's why you'll see throughout this talk, I'll refer to it as a hairpin, a pause, a terminator, but it's a hairpin structure. So once this structure forms, I want you to pay attention to this area here. Like you can see in this structure, when the RNA is single-stranded, there is a narrow opening to only allow single-stranded RNA. But for this structure to form, this opening has to open up to accommodate the structure, okay? And that opening up forces the polymerase to move extensively such that you see this part of the polymerase moving to the left by approximately 3 degrees. And that movement inactivates this polymerase such that it can no longer continue transcribing, and it's paused there for a long time. And if you were to compare the time, this was 15 seconds. You now have this pausing for approximately 2 minutes. That's how strong this structure forming here is. Now, with that said, I'd like to demonstrate that with my body just to make sure you get the point straight. So imagine me being RNA polymerase, and when I'm looking at you, when I'm actively transcribing, I'm looking at you straight up. But when this structure here forms, I'm now forced to take this weird conformation, okay? In this weird conformation, I cannot transcribe. I'm stuck here. But in this conformation, I can actively transcribe, okay? So when that structure forms, I'm in this conformation, I cannot move forward. That's gonna be important throughout this talk. I want you to remember that. So I'll again show you an animation for how that forms. You have an incoming nucleotide, you have a pause signal, and that allows that triggers elemental pausing, that allows the hairpin to start forming, and then you see the hairpin structure forms, and this is completely swivel. Now, with that introduction, what I actually was interested about was the role of a group of proteins called transcription regulators. What are transcription regulators? These are proteins that directly bind to RNA polymerase in order to regulate transcription, okay? And specifically, our group cares more about this protein called NusG. And the reason we decided to study NusG is because one, it is a conserved, it's the only conserved transcription factor in all domains of life. The second reason why we chose it is because this NusG is essential in most bacteria, including Mycobacterium tuberculosis that we care about, okay? But what exactly did we know about NusG before this project? So for one, we knew that NusG suppresses the pausing behavior that I just introduced in E. coli. And then the same protein, the same NusG enhances pausing in Bacillus subtilis. So I want to repeat this just to make sure it sinks in. You have the same protein that has completely opposite functions when you look at two different organisms, okay? So with that information, where we come in is we were asking, how does NusG, the same protein in Mycobacterium tuberculosis, actually affect pausing? Is it closer to this guy or is it closer to this guy, okay? And just to give you context, Mycobacterium has evolved approximately 3 million years. So this is over here. It's evolved approximately 3 million years for both Bacillus subtilis and E. coli. And so we didn't really know how its NusG might have evolved to affect pausing. And to answer this question, we used collaborative approaches like Bob mentioned. And these approaches included biochemistry, it included cryo-electron microscopy, and also included CRISPR interference, okay? And now I'll introduce how we used biochemistry to visualize these transcription events and actually interpret what's happening when we have the different NusGs. So here's the biochemistry assay. I made a schematic here to guide you on what could be happening. In this schematic, I'm focusing on three specific things. One is when the polymerase is paused. Two is when termination is taking place. And three is when RNA polymerase fails to terminate transcription, okay? At the paused state, you'll notice the polymerase is there. We still have the NusG bound to it, and there's the RNA hairpin. That is what I just talked about previously. And then at the terminator, we expect the RNA to be released and the polymerase to be released. But in the event that transcription termination fails to happen, we usually have this polymerase synthesizing this long RNA product. We call this the read-through RNA. So how does this three events look like in a biochemical gel? Well, one of the assays we use is called the time point assay. And as is in the name, we start transcription inside the tube. So we purified all these proteins and then start transcription inside the tube. And then after some time, we stop transcription and run it on a gel to see what happens. Now you'll see the time point goes from 0 minute to 1 minute, all the way to 15 minutes. But what's important is I want you to pay attention to the last lane, which is labeled C, or rather the control lane. So this control lane helps us interpret the event that could be happening at that particular moment, okay? And what I mean by that is I'd like to take you to this band, the high intensity band that you see over here. You'll see that throughout the time point, it's increasing in intensity, but then on the C lane, it disappears completely, okay? So that signifies that this is a paused RNA polymerase corresponding to this guy. And then when we add excess amount of nucleotide to this reaction, that pause escapes and extends further, okay? Now, the second band I want you to pay attention to is the lower band over here, okay? You'll notice similarly, this band is increasing, but then in the C lane, it fails to extend, or you still see it on the C lane. So this tells us that this is actually a product that's been terminated, okay? So the difference between this and this is a paused, you don't see it, termination, you see it in the C lane. So I want you to remember that because that's how I'm going to interpret all the data I'll show you today. The second assay that we used is called the endpoint assay, and this is slightly different from the time point assay in that we start transcription on a tube again, and then stop transcription after a specific amount of time, in this case, only 5 minutes, okay? In this case, we can only use this assay to look at termination events. We cannot use this for pause events. So this is more simpler than this guy. Now, using this assay, we did the first study where we compared the MTB NASG, Mycobacterium tuberculosis, compared to E. coli and Bacillus that we know their function. And these were the results that we saw. We could see that here without any NASG, you do see the pause, but it's very weak. When we add Mycobacterium NASG that we care about, you see this pause is enhanced or the intensity increases. This is similar to what we see with Bacillus, and as expected, E. coli suppresses the same pause, okay? So when we saw that this was interesting and we wanted to know more about it, and we actually decided to include even more NASG from even different species out there. And so we added another species called Clostridium difficile and another species called Bacteroidetes fragilis and looked at how they affect the same pause. So what we saw is you can see that there is a clear cut difference that NASG can either exist as a pause enhancer or it can exist as a pause suppressor. If you just look at the pause plays, you can see these are enhanced, this is suppressed. So this was interesting, and we wanted to know where's the difference coming from? Why would one NASG be an enhancer and the other one be a suppressor? So we picked 2 NASGs from each of these classes. One was our Mycobacterium tuberculosis and the other one E. coli, and then tested it using cryo-electron microscopy. And I'll show you how cryo-electron microscopy works. So we have the system that we care about with the polymerase, NASG, RNA hairpin, and it's all paused. And then we freeze this using a synthetic transcription bubble, okay? And once we freeze it, we visualize it on cryo-EM, and the data that you get from a cryo-electron microscopy looks like this. So these tiny particles, at this point, they don't mean anything, but what exactly they are is when you zoom in, you start seeing these 2D images that have different orientation. And the goal is to reconstruct all these 2D images to end up with a final 3D reconstruction here. In this 3D reconstruction, you can see in blue, this is our polymerase corresponding to this guy. In purple, that's our NASG corresponding to this guy. In red, that's our RNA. And in black over here, that is our DNA, okay? So using this setup, we were able to get a structure with Mycobacterium NASG and E. coli NASG side by side. And what we saw was interesting. So one, we had the no NASG as a control. Remember the no NASG has the hairpin, pause hairpin present, but it doesn't have any NASG. But you can see that the swivel state, the one I showed you, it's already swiveled because of the structure. And the non-template DNA, this is one part I also wanted to pay attention. We couldn't even see it at all. But then when we added E. coli NASG, then we start to see the polymerase being unswiveled. So remember, the swivel state was over here. When we add E. coli NASG, we saw it going back to this state, okay? And then when we looked at Mycobacterium NASG, then it stabilized the swivel state. So if the polymerase is over here, it sticks there. It actually becomes even more stable in this state, okay? Now, the other difference was in the non-template DNA. You could see that with E. coli NASG, we also could not see the non-template DNA, but we could clearly see the non-template DNA with MTB NASG present. So then we wanted to see how are these differences even possible? And we zoomed in to where we noticed a difference, okay? And on the left, I'm showing you the structure with Mycobacterium nasci and 2 parts of the RNA polymerase. Other parts are omitted for clarity. But these 2 parts, you'll notice MTB nasci is contacting RNA polymerase at this position here, and it's creating a space or a pocket that allows a non-template DNA base to flip into that pocket. This is in orange. This is the same base that I'm highlighting in orange. Now, if you look at the same position when E. coli nasci is present, you don't see that pocket. Why? Because E. coli nasci is contacting the polymerase at this position, as well as this position, shutting down the pocket that we saw over here, okay? So when we saw that, then we wanted to ask, does this interaction that we see here, that we do not see here, actually what's causing it to be an anti-pausing protein? So we zoomed in and identified the residue that are making interaction with RNA polymerase. And we identified this one residue, histidine 81 here, that's making the most extensive interactions. And when we mutated this to an alanine, all of a sudden you start noticing this pulse suppression effect of E. coli nasci, it's greatly reduced, okay? Suggesting that this contact here could be what's driving the pulse suppression effect, okay? So all what I've told you was in vitro on a tube. So we wanted to see, is this actually true in vivo inside a cell? And to do that, we made fusion proteins, and I'll try and walk you through this. So this is NASG and the different parts of NASG. And we have the NGN domain. There are 2 domains, the NGN domain and the cow domain. So we do know that the pause effect on RNA polymerase is isolated to this NGN domain. And when making our fusion proteins here, we only changed the NGN domain and left everything intact as the wild type. In this case, the wild type, we used Mycobacterium smegmatis because it's closely related to Mycobacterium tuberculosis. And when we made these fusion proteins, the goal was to see how does it affect the pause event, okay? So to make sure that we got everything right, we ran it side by side. You'll see here, these are the native NASGs that I previously showed you. We see pause enhancing and pause suppression. And when we use the fusion NASG, we also see the same properties here. So we were sure that it's only isolated to the NGN domain. So why did we do all this? That's where I'll introduce the CRISPR interference experiment that we used. So the way this works is we have a CRISPR system inside Mycobacterium smegmatis that we can control its expression of the dead Cas9, okay? So when we turn this expression, we overexpress dead Cas9 that targets the endogenous Mycobacterium smegmatis NASG. And when it comes and sits on this NASG, this operon is completely shut off. So the endogenous NASG is not able to be expressed, okay? How does this look like in a readout assay? So NASG is essential in mycobacteria. When you knock it down, you don't see any growth here. So, but when you don't have the CRISPR interference system on, you see normal growth. So the readout will be looking at how the different NASGs are affecting the growth of this organism, okay? Now, with that said, the way we will save a bacteria like this is when we turn on the CRISPR system, it's turning off the endogenous NASG, and then we introduce the NASG that we are interested to test. In this case, this is introduced at an ectopic site. So this allows us to test either a pro-pausing NASG or an anti-pausing NASG and see how it affects growth. So when we tested this side by side, we saw that the pro-pausing NASG had normal growth, similar to what we saw with the wild type Mycobacterium smegmatis NASG. But when we tested E. coli NASG, we saw poor growth. You can see a clear difference here. And what this results left us asking was, does this mean that the property of RNA polymerase pausing, is it essential in mycobacteria? So we hope to answer that someday, but right now we don't have the answer to that. Now, with that said, I'll summarize this part. So what I told you is the NUSG can exist as a pro-pausing NUSG or an anti-pausing NUSG. Pro-pausing NUSG stabilize a swiveled inactive RNA polymerase and context the non-template DNA. And anti-pausing NUSG stabilizes an active RNA polymerase and does not contact the non-template DNA. Okay. Now, with that said, everything I've talked about was part one, the interplay between elongation and pausing. I'll now shift my focus on what's happening during termination, okay? And I'm gonna take a water break as I let you digest that slide. Okay, so what happens during termination? In this case, we used a model bacteria, E. coli, as a model bacteria. We did not use Mycobacterium tuberculosis, because it's easier. It was easier to work with E. coli. And whatever results we found here is applicable to all bacteria, including Mycobacterium tuberculosis, okay? So one question you'd ask is, why will transcription terminate in the first place, or to begin with? And a few reasons that I highlighted here is to recycle RNA polymerase, 2, to prevent collision with other proteins, 3, to specialized RNA like tRNA need to perform their function, and 4, to turn off unwanted transcription for RNA degradation, okay? Now, for transcription to terminate, there are 2 major ways to terminate transcription, but in this talk today, I'll only focus on one of them and not the other. And the one I'll focus on is called intrinsic termination. So what's special about intrinsic termination is it relies on a structure that forms on the RNA, similar to what I just described, and this structure is called a terminator hairpin, okay? The way it works is once this hairpin forms, somehow disrupts everything, but that mechanism was not known or it wasn't clearly defined until our study. So what are the characteristics of this terminator hairpins? So it's a structure that resembles a hairpin, like I mentioned, but more importantly, this hairpin structure is followed by a series of 7 or 8 bases that forms a U-track. And more importantly, transcription itself must take place at these 2 positions, or one of these 2 positions, either at U7 or U8, okay? Now, just to make sure that you're not confused with the other structure that I introduced, I'm gonna highlight the differences between these 2 structures. Now, the structure that I talked in part 1 was a pause hairpin. It looks like a hairpin, but I wanted to pay attention to the distance from this hairpin to the active site. You have 11 bases in there, okay? For the terminator hairpin, if you look at the same distance, it has to be 8 bases. It cannot go past 8 bases. That's the main difference. Now, we use the same methods, biochemistry and cryo-EM, again, to look at what's happening during intrinsic termination, this time with the collaborators from Shanghai. And what we started with, the first experiment is, we knew that you need a terminator hairpin in order to terminate transcription. So the first question that we asked is, what happens if you get rid of that hairpin, or the ability of this RNA to form that structure, okay? And when we got rid of it and did the biochemistry assay and looked at the termination places, so these 2 bands over here, remember, if you look at the C lane, if you have a band staying that's terminated, if it disappears, it's paused. And what you can see here, there's a band that are accumulating, and in the C lane, it disappears completely. So that signifies that there is a pause event happening in the active site or in the termination site in the absence of this terminator hairpin, which suggested that the first step of terminating transcription is elemental pausing, or pausing briefly to allow the structure to form. Now, we repeated this. We used cryo-EM also to confirm that this was true, and used a scaffold or a synthetic transcription bubble with a scaffold that also lacked the hairpin. And what we saw here is, you can see in blue, this is the RNA that is bound to this transcription bubble. And more importantly, if you look at the active site, you can see that it's half translocated. similar to what we saw with elemental pausing. And just to remind you what elemental pausing looked like, here again, you have the incoming nucleotide, gets incorporated, then you have the pause signal, and only the RNA goes forward, the DNA stops where it was. So that's what we observed with that result. Now, the second thing we wanted to test is how does this terminator hairpin actually nucleate? Okay, and to the left, I'm showing you the native terminator, what we expect it to look like. But for the purpose of this study, we use a synthetic scaffold design in order to mimic this structure here. In this synthetic design, we used what we call an antisense RNA that base pairs to this single-stranded RNA over here. But more importantly, we did not include this last two bases. You'll notice this terminator hairpin goes all the way to the bottom. We did not include that here. That is because we didn't want the termination hairpin to fully form. We wanted to capture it while it's still nucleated, okay? And when we got the cryo-EM structure of this, it was incredible, actually. Here you can see in blue and pink, this is the DNA hybrid. And I wanted to look at the active site. You can see the active site. We also saw the half translocation that we saw with elemental pausing. There's nothing here, but there's something here, okay? But more importantly, if you go to the exit channel where we expect to see the duplex RNA, it was a very nice structure where we saw the duplex RNA forming over here. Just give it a minute. This is the duplex RNA. But I want you to pay attention to the surrounding polymerase, this part here. You can see when you start, it's narrow, like in pink, it cannot fit there. But then this part has to expand in order to allow this structure to fit there, okay? So when we saw that, the next experiment we tested is at what position does this terminator hairpin actually need to form in order to terminate transcription? And the perfect terminator in this case will be all the way to the right, where the antisense RNA here is going all the way to the bottom. And so we systematically deleted one base at a time, all the way to minus 13. It was actually minus 16. And then looked at the termination event taking place. And the result that we saw, remember you have to look at the C lane, is we start seeing termination taking place at the minus 12 position, with extensive termination taking place at minus 10 and minus 9, okay? So that will correspond to these two guys over here. Now, I wanna explain how the hairpin, terminator hairpin completes, okay? So this is our schematic. RNA polymerase is omitted. It's just the nucleotides. I want to show you how this RNA structure needs to complete forming over here. So initially, during hairpin nucleation of the structure that I showed you, there's two bases here, the minus 10 and minus 9, that are currently base pairing with the DNA over here, okay? But for these two bases here that are currently not bound to anything to bind to something, this have to release in order for them to bind to this guy. And so to the right, that will be what has to happen is the minus 10 and minus 9 DNA bases need to rewind or reanneal to each other in order to release the RNA on the back to complete forming, okay? So what if I told you that we can disrupt this process or this rewinding of the DNA that we see there? Because in normal transcription bubble, you usually have the bubble about 10 bases that are open. And then after transcription, this bases rewind and go back to the duplex DNA that we started with. But in our experiment, we decided to introduce mismatches at specific position such that when you rewind, these two places will not able to fully rewind because these bases are not compatible with each other. So when we looked at termination with these mutants here, you start to see that this is the wild type when everything is fine. You have a very nice termination efficiency, but the more you introduce mismatches, you completely wipe out this termination event. And more importantly, if you look at the minus 10 and minus 9 position, if we introduce a mismatch there, we completely get rid of termination. So what this told us is rewinding at this two position, minus 10 and minus 9, is required for termination to take place. Now, the other thing we wanted to test is during termination, what gets released first? You have the RNA, you have the DNA, and you have the RNA polymerase. And so to test that, we labeled the RNA and the DNA and then followed them over time, like what gets released faster, and then quantify that. And you can see in this experiment, down here, this will correspond to the RNA at the terminated site. And up here, this will correspond to the DNA. When we quantified this intensities, we saw that the RNA is released 5 times faster than the DNA. And the DNA is actually still bound to the polymerase, okay? And when we solved the structure for this, we were able to capture the RNA that has been released from the active site and is bound non-specifically elsewhere. And the transcription bubble and the DNA was still bound to the polymerase, okay? Now, what I want to finish with is what happens to the transcription bubble after termination takes place. And on the left here, this shows a normal transcription bubble during elongation where you have 10 base pairs that are single-stranded. But what we captured in the RNA release structure is this bubble was starting to collapse. It was rewinding and you could only see 5 base pairs that were single-stranded instead of 10, okay? So we were able to capture all these intermediate steps of termination taking place in bacteria. And to summarize all that in a cartoon form that I've been using for this talk is you'll see the incoming nucleotide that gets incorporated into the growing RNA. You have the pause signal. This allows elemental pausing. You have the hairpin structure forming, and then it completes forming. You have -10 and -9 reannealing. RNA is released, and then transcription bubble collapses, okay? And with that said, I'd like to finish by thanking the people, my collaborators, who without I wouldn't be able to learn the science and I wouldn't be able to show you the results that I showed you today. And I also like to thank my thesis committee, who my PhD depends on them today. So we'll see how that goes. And I also like to thank the Landic Lab and other labs that were part of this project by providing their input and all the useful discussions that we had with them. And lastly, I'd like to thank my friends and family who were really gave me the support that I needed to go through grad school and all the way until here. And lastly, I'm gonna take any questions if anybody has anything for me.
Great, so just to explain how the procedure works here for people who may not be familiar, this is the public part of Expery's thesis defense, and just gave that great talk, which I'm sure a few of you may have questions about. There's a period of time now when anybody can ask Expery questions, or the general audience, and then after that, the thesis committee will convene in private with Expery and complete the questioning. So, but the floor is now open for any questions that people have, and I'm sure Expery would be happy to answer them. So, questions. And if I can, I'm going to give you the mic, just because we're recording it, and that way we'll get the question.
Thank you, Expery, for this just a stunning, gorgeous, elegant talk. I was wondering whether you would care, so you discussed the role of swiveling in pausing, and then we've just had the conversation about termination, and I just was wondering if you wanted to speculate or send any thoughts about the role of swiveling in termination beyond the elemental pause, and if it would have any role in the rewinding event that you discussed. Thank you.
Yeah. So that's a good question. And you probably got it right. I didn't mention that with termination. So remember when I introduced swiveling, I said that for the structure, RNA structure to form, it has to be accommodated in the narrow space. So for any structure to form, that means the polymerase has to move. And that movement is what we call swiveling, okay? So for both pausing and termination, when the hairpin structure forms, the RNA polymerase has to take this conformation, or what we call swiveling, okay? So swiveling is important because during termination, for the hairpin, terminator hairpin to nucleate, the swivel has to stabilize it there to give it sufficient time for all the intermediate steps to complete forming until RNA is released. Does that answer the question? The way we think about it is swiveling is important to make sure the polymerase is stuck in that one position long enough for everything else to happen after. So we have not seen any proof that the swivel event itself is actually causing the re-annealing of the transcription bubble. So I cannot really speak to that, but we do know that swiveling does make the polymerase stay there for a very long time.
Other questions?
Excellent talk, Expery.
Thank
It was especially understandable for someone who doesn't study microbiology, so I can appreciate that. I was curious, are there similar mechanisms in humans if we were to turn this into a treatment for tuberculosis? Would you potentially disrupt transcription in human cells as well?
Yeah, that's a good question. So I mentioned that the protein NUSG is conserved in all domains of life, including humans, bacteria, and archaea. So in humans, there is a single study that suggested that human NUSG can either be pro-pausing or anti-pausing, depending on its phosphorylated event. But there's not been a lot of extensive study to really look at the mechanistic detail. So we do not know how that might be affected. But what we do know is in Mycobacterium NUSG, we know that it's pro-pausing. So if anyone was to show that human NUSG is anti-pausing, that is one difference that we can capitalize on and only focus on Mycobacterium NUSG without affecting what's happening in humans.
Thank you.
Any other questions? Could you pass this mic down all the way and I'll walk around?
Wonderful talk, Expery. I really enjoyed a lot of those visualizations as commenting on the previous person, because I really appreciate you walking us all through what you wanted us to learn. And so one thing I'm very curious about, considering how well you've really put this presentation together, I was wondering, after you complete your PhD, what's next in line for you? Because in terms of wherever you want to go, the one thing I really want to commend you on is how you really communicate this, because we need more people like you, especially regardless of whether you want to stay in academia or not, to really convey that excitement and connectivity with regards to science.
Oh, thanks. So that's a deep question. But I'm currently interviewing in a few places, industry and academia. And I'm waiting to see how those interviews turn out, and then I'll know where I'll end up. But it's good to know that you think I presented it clearly. Thank you.
So actually, we have one question that's been asked online. So we're going to read that to you.
Okay, this question is from Seth. For the termination assays, looking at the rate of RNA and RNAP release, were these done on linear DNA fragments? And if linear, what do you think the implications of that are?
Yeah, that's a good question. So I didn't touch on this, but this assay was done on a very short piece of DNA. Remember, I said that the cryo-EM scaffold design is synthetic. So we buy the DNA and we buy the RNA and assemble the transcription bubble in a tube. So the length of this DNA was about 50 base pairs, maybe 60. So very short, because in the cell, you have a long continuous DNA. So the results we are seeing here is this is 5 times faster in that short piece of DNA. This could mean that once the RNA has been released, the polymerase is sliding on this short DNA, getting to the end and just falling apart. So if we were to use a longer piece of DNA, like a circular DNA or a long DNA inside the cell, it's very possible that this RNA polymerase could be sliding on this DNA for however long, longer than at least what we are seeing here. So that's one experiment that we were thinking will be a follow-up experiment just to see how that compares to the results that we saw here. But good question.
Any other questions? Well, if not, let's thank Expery for a great seminar.